This method can help answer key questions in the bacterial pathogenesis field, such as when are host-killing virulence effectors expressed in bacterial cells and what are the virulence states of surface-attached and planktonic cells. The main advantage of this technique is that it is fast, quantitative, and the host cells are relatively easy to handle.
We first had the idea for this method when we noticed that surface-attached and planktonic subpopulations of Pseudomonas have different host-killing phenotypes.
To begin, grow E. coli strain BR overnight as outlined in the text protocol. Then, bring the E. coli culture to room temperature. Using a wooden stick, inoculate amoeba into 750 microliters of the culture. Immediately add 700 microliters of the amoeba E. coli mixture to a GYP plate. Tilt the plate to spread the culture evenly on the surface of the plate. Then, place the plate in a box that maintains high humidity. Incubate the plate inside of the box at 22 degrees Celsius for 4 to 5 days. After the incubation period is over, use a sterile pipette tip to collect amoeba spores from half of the plate. Then pipette up and down to resuspend the amoeba on the pipette tip in 1 milliliter of PS medium. Transfer the inoculate mixture to a 250 milliliter flask with 25 milliliters of PS medium supplemented with antibiotic antimycotic solution. Grow the amoeba at 22 degrees Celsius in a rotating shaker at 100 RPM. After